Okay, so rather than just talk about the insides of cells, why not bring a coronavirus into this? And the reason why I'm doing this is that this is the area where I actually work. And so these are the images that I have, and I thought, why not bring you some actual scientific images? And you can look at these and interpret them right along with me. So, what do you see here in this big blob in the center? You see those dark gray things, squiggly things? Can you figure out what those are? I see a lot of stripes, and the stripes are generally running this way, although this one we caught at a weird angle, but here you can see them there, there, and there. Those stripes look very slimming to me, right? Those are mitochondria, and these are pretty healthy mitochondria. And you can just start to see the edge of this giant thing down here. It's usually going to have dark material right lined up, basically where the nuclear pores are. This is the nucleus, and this is just a little piece. The entire nucleus would be, about as big as this entire image, probably. And then the other things you can see in here are the little rings, like little greasy gray donuts. Those are actual coronavirus particles. And so this is going to be SARS coronavirus. And these little chambers up here with this sort of squiggly stuff inside, those are where the SARS coronavirus makes its copies of itself. So these are its little replication factory that it builds inside your cells. Pretty neat. All right, so let's talk for a second. Every kind of virus that has RNA inside of it instead of DNA inside of it, every kind of positive strand virus, but let's just say every RNA virus for now, is going to make some version of a little house. And the house is going to have some way that you can get materials into it. It's going to have some way to get materials out of it. And it's going to provide some protection while the virus is copying itself. So viruses, you can think of them as being kind of shy, but really it's just that there are a lot of things in the cytoplasm that would try to detect a virus and would try to shut it down if they could find it. And so viruses will take over different parts of the cell. They'll steal the endoplasmic reticulum, or they'll steal mitochondria, like this guy over here, flockhouse virus of, what does that infect? I think it's a tick-borne virus of chickens, if I remember right. Rubella virus over here, great. I think that one is German measles, although I don't know. I'm not so good at my names outside of coronaviruses. Here's poliovirus. It's going to make all these little things out of what used to be the Golgi apparatus. And some viruses will take over both the ER and the mitochondria. Look at that. Clusterovirus. These infect plants. There's a really bad one that infects orange trees. It's called citrus tristeza, like citrus sadness virus. Okay, so each of these things that we just showed is actually made of membranes. And so it turns out that all of these RNA viruses are very, very dependent on the membranes of the cells. And the membranes have to be in motion. They have to keep being made and keep being broken down, or else viruses can really suffer. And so there's a big enzyme called the fatty acid synthase that has each of these little parts. And so they're showing you the structure of the parts that we've actually solved. But each of these little parts is doing a different step in the reaction to actually build a fatty acid molecule, which we can then attach to make like a membrane lipid, or we can attach it to glycerol to make a fat molecule like a triacyl glycerol. And that fatty acid synthase molecule, there are all these different little parts to it. So we can count them up. 1, 2, 3, 4, 5, 6, 7. So 7 different parts that actually do 7 steps of the process. And the neat thing is you can go in and inhibit different parts of the process. So one of the chemicals, and the one that we're going to use is this one right down here. It's called C75. And basically you need this entire assembly line to be working in order to make fatty acids. And if you stop the assembly line at any point, then you are just absolutely preventing fatty acids from being made. There's no way out of this. And so they're showing you some of the molecules that actually act as inhibitors of different parts and putting to the part. But C75 is the one we're going to focus on here. And so you'd say, all right, well, what does C75 do to a cell? And so here we're looking at different concentrations of C75, and we're looking at the amount of cell growth. It's a thing called an MTT assay. It's actually looking at the amount of cell metabolism that's going on. And you can see that for the ones that are treated with C75, it's about the same as for the ones that are treated with the stuff that C75 is dissolved in. It's a low amount of a chemical called dimethyl sulfoxide. And so you can see the dimethyl sulfoxide is a little bit toxic. It's driving that number down. But the C75 isn't really anything different than no C75 there. So it's not killing the cells, but look what it does. Look what it does to the virus. So here the blue line is, these are different concentrations of C75, or just the stuff that C75 is in. We call that vehicle. That's the dimethyl sulfoxide mix. And you can see at high amounts of C75, the virus is really low. And at low amounts, the virus grows just fine, or rather, when it's not present, the virus grows just fine. And it's actually the same level of virus growth as when there is very low amount of C75. So yeah, it stops the virus from growing, which is pretty neat. And let's see, it also stops the virus from accumulating. So as the virus grows, it's going to make a thing called double-stranded RNA, because coronaviruses have RNA inside instead of DNA. So they don't have DNA at all. And when they want to copy themselves, they'll take their RNA and make another RNA copy of it, which makes a double-stranded RNA. And so you can look at this stuff appearing, and basically if you treat it right when the virus is coming in, then it really hits it hard. If you pre-treat it, it doesn't hit it all that hard. And or rather, if you put the virus in before the treatment, the virus is actually fine. The C75 works at the level of the virus getting going. It basically works at the level of the virus making those factories, which is kind of neat. So I'm telling you all of this because these are the images that you are going to be looking at. You're going to be looking at electron microscope images. And I want you to have the rest of the story there, just so you understand what's going on. Here you can see the nucleus is shown in blue. The little dots are showing fatty acid synthase. Everywhere there's a little dot, there's a fatty acid synthase molecule. And everywhere that there's red, there is double-stranded RNA. So you can see there's the nuclei are here. They're just a little bit fainter. They're a little bit bleached out, but you can see the nice little double-stranded RNAs. Looking great. Okay, so you will see some amazing things inside of these cells. So these are some representative C75 treated cells, and boy do they look weird. But before you can understand what C75 treated cells look like, you need to see what regular cells look like. And so I think that's a good place for us to go to a different different lecture. So we will leave off this one, and then these images, most of these are going to occur in the third presentation that I do. So this has been the end of the introduction, and now let's go look at the insides of cells and figure out how they work.